Well, good evening, everyone, and welcome to the last Fralin Biomedical Research Institute Maurice Strauss Distinguished Public Lecture of 2021. We have the holiday break coming up, of course. And just a quick reminder before I introduce tonight's speaker that after the holiday break, the next installment in our series will be on January 20th, and that'll feature Dr. Sharon Peacock, Professor of Public Health and Medicine at Cambridge University, where she also serves as the Executive Director of the COVID-19 Genomics Consortium of the United Kingdom. And Dr. Peacock will be speaking on SARS-CoV-2 variants, past, present, and future. Obviously, a very timely topic for everybody. So without further ado, it's my pleasure to introduce this evening's speaker, Dr. Diana Bianchi, Director of the Eunice Shriver National Institute of Child Health and Human Development. Dr. Bianchi is also head of the Prenatal Genomics and Therapy section for the Medical Genetics branch of the National Human Genome Research Institute. As Director of NICHD, Dr. Bianchi oversees the research on pediatric health and development, maternal health, reproductive health, intellectual and developmental disabilities, and rehabilitation medicine as well. She received her undergraduate degree from the University of Pennsylvania and her medical degree from Stanford School of Medicine. She did residency training in pediatrics at Children's Hospital Boston, and then postdoctoral fellowship in medical genetics and neonatal perinatal medicine at Harvard. She previously worked at Tufts Medical Center, where she founded the Mother-Infant Research Institute. Dr. Bianchi has authored numerous scientific articles and co-authored the book on diagnosis and management of the fetal patient. She also serves as editor-in-chief of Prenatal Diagnosis and has been the past president of the International Society for Prenatal Diagnosis and the Perinatal Research Society. Dr. Bianchi is an elected member of the National Academy of Medicine. She's also a recipient of many awards, including the Colonel Harlan D. Sanders Lifetime Achievement Award in Genetics from the March of Dimes for her work on maternal and fetal cellular communication, and the Landmark Award from the American Academy of Pediatrics for her work in genetics and newborn care. Her own research is focused on prenatal genomics and non-invasive prenatal DNA screening and understanding the fetal transcriptome to develop new therapies for genetic disorders that potentially can be given prenatally. Her labs worked on fetal and placental biology through sequence analysis of nucleic acids that circulate within the pregnant mother's blood to help improve counseling and prenatal care. She's also studied the effects of maternal obesity on fetal gene expression, developing novel prenatal therapies, and has done a lot of work in Down syndrome, of course, studies of the amniotic fluid transcriptome, where she and her team have found that cell-free RNA in human mid-trimester amniotic fluid supernatant can contain multiple fetal brain transcripts. So they're developing exciting new approaches to prenatally treat the stresses such as oxidative stress or inflammation in the nervous system in fetuses with Down syndrome to potentially help improve neurogenesis, synaptic plasticity, and thereby improving postnatal cognition and behavior. So without any further ado, I'm going to introduce her now and say we're going to hear from Dr. Bianchi this evening on the subject of transforming prenatal screening from new technologies to gender reveal parties. Please join me in welcoming Dr. Bianchi.
Thank you so much, Dr. Friedlander. I'm so glad we could finally do this. We've been talking about it for a long time, and kudos to whoever did the research on my background. So somebody was looking at our publications, which is great. So let me just share the screen, and hopefully everything will work as it should. Get into presentation mode. There we go. Hopefully it is in presentation.
Yep, you're good. We can see it just fine.
Wonderful. So thank you. Don't jump ahead. Thank you for the kind invitation. It's a real honor to give the Maurice Strauss Distinguished Public Lecture. And of course, I have two roles, as Dr. Friedlander mentioned. It is my great honor and privilege to serve as the 8th director of NICHD. And obviously, I'm the first woman, but that's not why I made the slide. I made the slide because one of our senior program officials once said to me, you are the first director of NICHD to have a full head of hair. And I wasn't sure if that was true, but through the NIH library, I did get pictures of all of the previous directors. So it is true. And I also just celebrated my 5th anniversary. So I feel like finally I have arrived and I understand what it is I'm supposed to be doing. And of course, today is a very emotional day because tomorrow essentially is Dr. Collins's last day. And so we've been listening to tribute videos and it's been a very emotional time for us. But today I'm going to be talking in my other role as a senior investigator. And I know that you're a research institute and I love research. And that's why I am so privileged to be able to not only do research, but to see it implemented into clinical care. So what I'm going to be talking about is something that my colleague, Eric Green, who's the director of the National Human Genome Research Institute, said a few years ago in Genome-Web. He was talking about prenatal genomic testing, and he said that it's probably the #1 implementation of genomic medicine. And he's correct. It's still correct today. So I'm going to give you a little background. I know there's a very mixed audience here, so hopefully it'll be something that everyone can find accessible. I'm going to talk about the clinical implementation, and then what we call secondary genomic findings, which make research so interesting because we never dreamt that we would be finding things that we find as a result of this seemingly routine testing. And then I'm going to end because I think it's important to think about societal implications and some of the unintended consequences of this testing. So as Dr. Friedlander said, I had the privilege also of being the editor-in-chief of the journal Prenatal Diagnosis for 13 years. And as part of my swan song there, I did an analysis of what the journal had published over its first 40 years. And you can see that some fields kind of ascend and then flatten and then descend. The field of prenatal diagnosis originated in 1966 when amniocytes were first successfully cultured from amniotic fluid, but it was really not until 1980 where it was really beginning to be a separate field in medicine. And the main technique at the time was an invasive diagnostic technique, amniocentesis, to get a sense of what do the fetal chromosomes look like. And that is that pink line up at the top of the graph. The testing from maternal blood began around the same time in the early 1980s, when it was recognized that fetuses that had open neural tube defects released the protein, alpha-fetoprotein, into the mother's blood. And that really started this whole field of non-invasive screening. When it was recognized that fetuses with neural tube defects had high levels of alpha-fetoprotein, but fetuses with Down syndrome also had abnormal levels of alpha-fetoprotein. What I'm going to be talking about today is this yellow line that really doesn't get started until about 2011, but it has eclipsed most other forms of testing quite rapidly. And this is another snapshot from the paper, just sort of looking at in PubMed, medical categorization of articles showing that in the 80s and 90s, one of the big areas that was being looked at with serum screening was Down syndrome. At the time, biochemical screening and ultrasound screening were the predominant ways of screening. But what happened in 2008 was that massively parallel sequencing machines became affordable enough that individual laboratories could afford them. And there was a recognition, more or less at the same time, by Stephen Quake on the left at Stanford and Dennis Lowe on the right at the Chinese University of Hong Kong, that you could use these massively parallel sequencing machines to sequence fragments of DNA that were floating in the blood of people with cancer and women who are pregnant. And you could use the sequencing not to determine a base change in the DNA, but you could use it as a counting system. And so the hypothesis was that if you had extra amounts of material from chromosome 21, for example, you could use the sequencing to screen for Down syndrome. Now, there are two main methods of screening by what we call non-invasive prenatal testing. One is random genome-wide sequencing, and the other is targeted sequencing. And I like to think of it as either fishing with a huge net that you throw out and you pull up the net and you get everything that's in the net. So that refers to the cell-free DNA that is randomly sequenced, it's mapped to specific chromosomes, and then it is counted relative to a reference to identify pregnancies at risk for aneuploidy. Like I said, if you find extra amounts that map to chromosome 21, that would suggest that the fetus has Down syndrome. Usually the reference chromosome it's a different chromosome. So if there's a problem in the other chromosome, it might artificially make you think that there's something wrong with one of your chromosomes of interest. Many of the bioinformatics algorithms only are looking at the chromosomes of interest, generally chromosomes 13, 18, 21, the X and the Y. But you can open up the results to see what's going on if you get unusual results, which we'll be talking about later on. With targeted sequencing, it's more like fishing with specific bait, where the bait is going to pull up segments from the chromosomes of interest, chromosomes 13, 18, 21. And in those types of techniques, you're using the mother's DNA as a reference, and you're looking at differences in the amount of DNA in what you're extracting from the plasma, which has the fetal DNA, compared to the mother's white blood cell DNA. So it's important to recognize that when I say fetus, I really mean placenta. The DNA that is circulating in the blood of the pregnant woman is really from the placenta, not from the fetus. It's used as a proxy of the fetus. And both the mother and the fetus produce cell-free DNA from cells that are undergoing apoptosis. If we were live in person, I could look at all of you and tell you that you all have circulating DNA in your plasma. But if you're an adult and you're not pregnant and you don't have cancer, your DNA is coming from your bone marrow, and it's released as your blood cells undergo recycling, essentially. Again, the fetal DNA originates in the placenta. And when we take a blood sample from a pregnant woman, we are getting a combined blood sample that on average is about 90% maternal DNA and 10% fetal DNA. And you sequence them together. So you don't really know what's the mother's DNA and what's the placental DNA, except if it's a male fetus. You can detect the Y chromosome, and in most cases, except for rare genetic unusual conditions, that means that there's at least one fetus in there who is a male. And you can also detect unique polymorphisms or variations in the DNA that are inherited from the fetus's father. And we've also done experiments in collaboration with Graham Burton's laboratory at the University of Cambridge in the UK to actually prove that as the placenta, or in this case, the syncytial trophoblast cells of the placenta undergo apoptosis, they release DNA into a tissue culture medium. So we've not only shown that oxidative stress can create damage to the placenta, releasing DNA, but you can reverse that with certain antioxidant vitamins. So we know that as the placenta is turning over, and if the placenta is undergoing any sort of stress, it's releasing cell-free DNA into the mother's circulation, and the mother's liver is metabolizing it, and it's eventually excreted through her urine. So what about the clinical implementation of NIPT? Well, this is a screening test, and it's very important to recognize the difference in screening versus diagnosis. So a screen is really meant to detect people who are at higher risk within the general population of a particular condition. When we started out, it was originally offered as an alternative to invasive diagnostic procedures for women who are at high risk for fetal chromosome problems. So who is at high risk? Women who are over age 35 when they deliver, fetuses that have a structural abnormality in one of the organs on ultrasound, a woman who's previously had a child with a chromosome abnormality, and then a woman who's had other abnormal screening tests, which are largely biochemical tests. More and more, because as I'll show you, the test performs so well, it's being increasingly offered as a first-tier test for all pregnant women. But if you get a positive screen, professional societies uniformly recommend, I say require here, that's what it says in the policies, but most women don't necessarily have a diagnostic confirmation with either an amniocentesis or chorionic villus sampling, which are procedures that get fetal or placental tissue. And what we're going to talk about a little later on are the false positive cases or the non-reportable cases in which you get unusual results that are either non-reportable or abnormal, and then you get a fetal diagnostic test, and that shows that the fetus is actually normal. So what do you do in that circumstance? So we will be covering that. Now, the testing really became clinically available just 10 years ago. So really, I'm talking about something that has come into clinical practice within the last 10 years. And the reason why it has taken off is that the positive predictive value, which I have circled in red, is so high. So for women who are at high risk, I told you what makes you high risk earlier. Women who are at high risk for having a fetus with Down syndrome, if you get a positive screen with this non-invasive prenatal testing, there's a 91% chance that the baby actually has Down syndrome. That's a very, very high positive predictive value for a screening test. And the negative predictive value is extremely high. It's over 99%. You can see that the risk is 1 in 1,000. So that's important to keep in mind when we talk about the downstream effects of the test. Now, there are 2 other major trisomies, and the reason why we focus on trisomies 13, 18, 21, they are the major live-born trisomies that are associated with major congenital anomalies. So trisomy 13, for example, the older testing of serum biochemical proteins and ultrasound really doesn't even detect that it's not screened for regularly. And women at high risk, the positive predictive value for both trisomy 18 and trisomy 13 are 84% and 87% respectively. Well, that's for high-risk women. When the testing first became available, the question was, okay, those are women at high risk. What about women of general risk or low risk? And so there were 2 studies, one of which we did in collaboration with Illumina, and the other was done by Mary Norton's group at the University of California, San Francisco, in collaboration with a company called Ariosa at the time. And we took women and compared them to the standard of care, which today is still biochemical testing with ultrasound testing, versus the DNA testing. And this is very early on. So this is 2014, and it's actually much better now. And the positive predictive value for the current standard of care, the biochemical testing plus the ultrasound, is only on the order of 4 to 5%. It was 3.4% in the other study. That means that there are a lot of false positives. So a lot of women are having unnecessary testing. The positive predictive value in our original study was 62%. As I said, it's much higher now, even for general risk women, more on the order of around 90%. And the negative predictive value is greater than 99%, which means that if you tell a pregnant person that there's a greater than 99% chance that her fetus does not have trisomy 13, 18, or 21, a lot of women say, that's great. I am not going to go further. I'm not going to have a needle put into my womb to find out about the fetal chromosomes. But we'll talk about the downside of that. So what happened from 2011 and continues to this day, if you see in blue, this rapid increase in the number of tests per year, as that went up, the number of diagnostic procedures compared to 2011, before the testing became available, began to come down. And as I said, because of these very high negative predictive values, many women did not want to have a diagnostic procedure. And so what's happened over the last 10 years is gradually this has become incorporated into practice. Commercial insurance largely covers testing for all high-risk pregnant women, and more for women at general risk. I wanted to show this picture because Virginia is in green, and this relates to state Medicaid programs. Now, Medicaid pays for care in just about 50% of pregnancies in the United States. And I find it surprising that there are even 6 states that cover with Medicaid insurance non-invasive prenatal testing for women at average risk. But there are also the red states here, doesn't mean anything politically, but there are red states that do not cover non-invasive prenatal testing under any circumstances. And the majority of state Medicaid programs will cover it for high-risk pregnant people. So over the last 10 years, there's been this rapid incorporation. There have been globally well over 10 million tests performed. And as I've said, it's resulted in a 50 to 70% reduction in diagnostic procedures. So it's a thumbs up, because this is great. It's safer for pregnant women, particularly women who've had a hard time getting pregnant, who are older, who might be at a higher risk of miscarriage. There is no risk of a procedure-related miscarriage if you're just having a blood test. However, there's been a lot of confusion about what you're testing for when you do this test. So you're only screening for chromosome abnormalities, either whole chromosomes or parts of chromosomes. And if there is a single gene disorder in the family, let's say the family has a positive family history of cystic fibrosis, the test is not going to look for cystic fibrosis or any other single gene disorders. And there seems to be a lot of confusion about that. Also, because there's been this massive reduction in procedures, trainees do not learn how to do these procedures. So there are attempts to make models or have, I'm blocking on the word, but where they make an artificial uterus and teach people how to do the procedure. Simulation, that's the word I was looking for. Simulation. But it's not quite the same as learning on a real live person. And because there are significantly lower test volumes in cytogenetics laboratories, this has resulted in the reduction of a significant number of academic cytogenetics laboratories. So it's not all good news, although it is safer for pregnant people. Well, one of the things we're going to be talking about at the end, the unintended consequences, is the role of industry. So what has made this journey somewhat different from other academic tests that made their way into clinical practice is that from the beginning, industry played a significant role. And full disclosure, at one point, I did have my research funded by Illumina. And there were pluses and minuses to that. They never limited what I could publish. And it did give me access to an enormous clinical laboratory for the purpose of research. So on the whole, it was a very positive experience and certainly gave me experience in public-private partnerships, which is something that we are exploring at the NIH today. However, the fact that industry was so involved from the beginning certainly changed the way this testing rolled out, which is why I think this is a very interesting topic for a general audience. It wasn't even recognized that cell-free fetal DNA was in maternal plasma until 1997. And the first detection, not by sequencing, but by PCR, of increased amounts of chromosome 21 sequence wasn't until 2006. I already explained that it was really 2008 where the technology became available. And between 2008 and 2011, my laboratory, among others, were busy doing clinical studies to see how accurate were these screens. And they were shown to be quite accurate. We started with trisomy 21, but it became clear that 13 and 18 were also quite accurate. And that led to a rapid incorporation and competition between companies. So by 2012, companies were already offering sequencing for the X and the Y chromosome. And of course, that proved to be enormously popular because it was a way that with a blood test, you could tell whether you were having a boy or a girl. And that brought up certain issues because we never screened for fetal sex by DNA. Of course, by ultrasound, you can tell fetal sex, but it also led to screening for some of the sex chromosome aneuploidies, or the SCAs. So conditions where boys might have an extra X chromosome, like Klinefelter syndrome, XXY, or girls are missing an X chromosome, Turner syndrome. We had never prenatally screened for those conditions before. So there were a number of ethical concerns as to whether it was appropriate to do that. Those conditions, if anything, are associated with very mild learning disabilities. There can be some mild congenital anomalies, but for boys with Klinefelter syndrome, for example, generally they don't have any congenital anomalies. They look absolutely typical compared to any other child, but they will have infertility later on in life, for example. So this brought up all kinds of questions. And then because the companies were trying to distinguish themselves from each other, they began to push the envelope. And because they could detect large areas of deletions of certain chromosomes, like, for example, DeGeorge syndrome, which is associated with a whole segment of DNA that is deleted from the long arm of chromosome 22. And then there were other conditions. At least DeGeorge syndrome occurs about 1 in every 4,000 people, but there were other conditions that were named in the microdeletion panel, like Jacobson syndrome, that only occur 1 in 100,000 live births. And in my practice of medical genetics, over 20 years, I've never seen a patient with Jacobson syndrome. So we are screening for that because we can on a technical basis. And so the rest of the timeline talks about the expanded testing menu. NIPT for rats we'll get to in a moment. Those are rare autosomal trisomies. And the beat goes on. It is increasingly being incorporated as a first-line test. In 2018, Dr. Rosa Chu from Hong Kong and I wrote a review article that is still pretty much up to date today that talks about what what is offered as part of current obstetrical practice. It's the common trisomies, as well as the sex chromosome aneuploidies. Women have to pay extra for the rare autosomal aneuploidies, as well as these microdeletions that I mentioned. And one iteration of the test is looking all across the human genome and looking for smaller and smaller variations in the DNA. Well, remember I said the testing first became available in 2011. By 2013, there were reports that this test was very inaccurate. There are false positive results. The test says trisomy 13, the fetus has normal chromosomes. There was concern that women were ending their pregnancies prematurely for a screening result without getting a diagnostic test and without finding out that the fetus was actually normal. There was a lot of concern, as shown in this paper, that perhaps it was time to sound an alarm about these false positive test results. With the benefit of hindsight and a lot of experiments, we now know that the test is actually quite accurate from a technical perspective. It's just more sensitive. And so there are a number of different biological reasons to get an abnormal screening result that are perfectly legitimate, but they're not necessarily coming from the fetus. And the most common is probably confined placental mosaicism. So I have a nice picture of a placenta here for those of you who haven't seen a placenta. In this case, we're looking at the inside of the placenta. They're actually membranes, and the fetus would be inside this with the umbilical cord, which is that pale yellow thing, attached to his or her belly button. And the confined placental mosaicism is not uncommon. And what it means is there's a little segment of the placenta that has abnormal chromosomes. And that happens very early in embryonic development. And as that section of the placenta is turning over, it's releasing abnormal amounts of DNA of a particular chromosome into the maternal circulation. So we now know that that can be detected via this technique. Sometimes the fetus itself has mosaicism, meaning it's got part of its chromosomes are normal, part of its chromosomes are abnormal. We can also detect the fact that there may have been twins there, or there's a rare autosomal trisomy. So let me just quickly go through that. We didn't realize at the beginning that some of these abnormal results could be due to rare autosomal trisomies because the results were masked, And so we collaborated with a laboratory in Australia that was using similar techniques to what we were doing. And we essentially opened up the whole genome sequencing results. And what we found in our group, as well as a collaborating group in Australia, that we could explain some of these false positive results by the presence of rare autosomal trisomies, either in the placenta or the fetus. And there were certain chromosomes for which it was more common to find a rare autosomal trisomy. But since the conventional wisdom, like when I trained in genetics, we were always told that if you had trisomy 7, you would not make it through the pregnancy. You would miscarry that pregnancy. Similarly for trisomy 16 or 22 or 3. But what we found is that actually a lot of these pregnancies are continuing to term, and we never knew that there was a rare autosomal trisomy. And it turns out that one of the biggest and most important conclusions of the study was finding out that if you had trisomy 15, which as you can see here is the second most common of these rare autosomal trisomies, that you were more likely to miscarry in that pregnancy. Conversely, if you had trisomy 7, NLB here means normal live birth. So with outcome data, we were able to understand a little bit more about what was driving these unusual results, and then what did it mean in terms of counseling the patient. And a lot of people said, well, does this have any clinical utility? And we argued, yes, it does, because it identifies cases at high risk of miscarriage, also would explain a miscarriage if it occurred. And we actually changed management recommendations. So if a rare autosomal trisomy was detected, we recommended that women should have an ultrasound for fetal viability, and that there were 2 chromosomes that if we found it, it was associated with a high rate of miscarriage, even after it looked like the fetus was alive on ultrasound. And there's something called uniparental disomy, where initially you have 3 chromosomes, 2 from one parent, 1 from the other parent. And if you lose the 1 from the other parent and you're left with 2 chromosomes from the same parent, that can cause a problem if the genes are imprinted and you need to have a copy from a mother and a father rather than 2 from the same parent. We also found with our outcome data that rare autosomal trisomies are important to know about because they are associated with fetal growth restriction, a propensity to preterm birth, and also a slight increase in congenital anomalies. So this was the first kind of finding that we really didn't expect, because we didn't think that the test was going to detect anything other than the common trisomies. But the bigger issue was recognizing that a lot of these abnormal screening results were due to the mother. So remember that I told you that 90% of the circulating DNA is actually from the mother, and around 10% is from the fetus or the placenta. And the testing itself will recognize variations in the mother's DNA. It also will recognize if the mother has mosaicism for a sex chromosome aneuploidy, most commonly 1 X chromosome instead of 2 X chromosomes. So it's very important to recognize that if there's an abnormal result, it is not necessarily the baby. We also now know that certain conditions that the mother has will affect the quality of the DNA, and that affects how the sequencing works and can give false or abnormal results. But there have been some really interesting cases. I get a lot of virtual consultations. And a common scenario is, you won't believe this, but it is a relatively common scenario. The DNA result comes back and it says normal male fetus because the Y chromosome is detected. And then the pregnant woman goes on to have an ultrasound and the fetus is clearly female on ultrasound. So how can you have a Y chromosome and a female fetus? And people freak out. And then it turns out, and this has happened more than once, that the mother has a history of an organ transplant, most commonly a kidney transplant from her brother or a male relative. And there's always micro levels of organ rejection that result in a release of Y chromosomal DNA. And that is more than what is coming from the placenta. So it outcompetes essentially the DNA from a female fetus and artificially makes the result look like it's a male fetus. Now you would think that the obstetrician would know about that, but and maybe he or she does, but they don't necessarily put it into the testing report. So the test is just measuring the DNA, doesn't know that there's been a previous organ transplant. And then fibroids, which are generally benign tumors, are also detected by this. But the result that really has inspired secondary research for us at the NIH is maternal malignancy. So again, back in 2013, around the time that people began to recognize that there were false positive cases, there was a case report of this woman in North Carolina who on multiple tests had evidence of several abnormal chromosomes. And here she is. Unfortunately, she has since passed away. But when she was pregnant with her son, she did have the non-invasive prenatal testing, and the test was repeated 3 times, always giving the same abnormal results. And it wasn't until after she delivered that they recognized that she had a tumor and it was widely metastatic. And she was first detected with pathologic fractures because she had pelvic pain after delivery. And she turned out to have a neuroendocrine tumor that was widely metastatic. She was treated and lived for 7 years, but she was the first person to be recognized, having cancer being detected by non-invasive fetal testing. And since then, we and other groups have done large studies pulling from these very large scale commercial laboratories, looking at the non-invasive prenatal testing samples, and then looking retrospectively at the patterns of the DNA testing that these women had, because we knew from the outcome data that they had unusual results and their physicians reported that they had cancer, and they reported back to the lab, could this be cancer? And then we, at least in our study, we reconsented the women to look at all of the chromosomes, because the women were at the time just signing permission to look for trisomies 13, 18, 21, the X and the Y. So you can see the numbers are not large. And this is really looking through the lens of a test result that has multiple aneuploidies. You would not see multiple aneuploidies in a living fetus. So you know that when you see a test result with multiple chromosome abnormalities, something is profoundly wrong. And so many of these women did have cancer, or they had fibroids. And most of the time it was a solid tumor, but some of the women had leukemias. And I've been, up until fairly recently, I was keeping track and kind of had a monitor of about 100 cases, what were the proportions. And the majority of women, these are young women now, the majority of them have lymphoma, which is a tumor that you would expect in young pregnant women. Colorectal cancer is also fairly common. And it depends where you are. So in the United States, for example, you're much more likely to have lymphoma or colorectal cancer or breast cancer. If you are in Asia, you can have nasopharyngeal carcinoma. Not all of these cancers are reported in the literature. It's becoming more increasingly recognized. And in lecturing to maternal fetal medicine providers, I estimate that about 1 in 5 have already had at least one case of cancer detected in their practice. And you may know about liquid biopsies because you've heard about liquid biopsies, and there are tests, screening tests that are designed to look for cancer in asymptomatic people. And just to point out that this test was not designed to detect cancer, but it does. So before I came to the NIH 5 years ago, I really, really wanted to do a study prospectively to find out, what should you do in these cases? When should you do additional testing? When should you wait? When should you worry? The problem is that insurance companies will not cover a major maternal workup because there's no evidence. So we have to gather evidence. In fact, even people who practice prenatal genomics debate whether you should report on results that suggest that there might be maternal cancer. There's no evidence-based guidelines. And even if you know about this and you're an obstetrician and you refer your patient to an oncologist, most oncologists don't know about this. They see a healthy, asymptomatic pregnant person, and they say you have nothing to worry about. The other problem is the test reports are quite different from each other. So, and there are multiple laboratories, especially in the US, offering this type of testing. They all report the results differently. And the patients themselves are reassured by the fact that they feel well, they're happy to be pregnant, they don't know what the fuss is about. Maybe their doctor will order a chest x-ray or some limited evaluation, and if it's negative, they're falsely reassured. So for the last 2 years, we've been fortunate to be starting a study at the NIH Clinical Center, America's research hospital, which is shown in the picture, the largest research hospital in the world. And the identify acronym is standing for Incidental Detection of Maternal Neoplasia through Non-Invasive Cell-Free DNA Analysis. So our work through NHGRI is in collaboration with the National Cancer Institute, as well as the Clinical Center, the Department of Radiology, and the Department of Bioethics. And our objectives are to collect prospective evidence to determine the best approach for diagnostic workup of pregnant women who receive either false positive or non-reportable NIPT results. We also want to identify DNA sequencing patterns and other biomarkers that correlate with the presence of a maternal malignancy. And basically, we're providing proof of principle for the liquid biopsy, even though the test isn't designed to do that. So women can participate if they're over 18 years old and they've had an abnormal or a non-reportable NIPT result. We ask that there is evidence that the fetus is viable and that looks normal on ultrasound examination, or that there is a normal diagnostic test result, normal chromosomes, normal chromosome microarray. And we're enrolling pregnant women or up to 2 years postpartum. I'm just going to quickly give you a couple of examples of cases that have been part of the study so far. We started right before the pandemic, almost exactly 2 years ago in December of 2019. The first woman was 32 years old in her 7th pregnancy, in the 3rd trimester, 30 weeks. And the reason why she ended up having non-invasive prenatal testing was on a fetal anatomy scan, there was a fetal cardiac abnormality. I don't know why Fragile X testing was ordered. There was no indication for that, but the indication was to do an amniocentesis to look for chromosome abnormalities. The fetus had normal chromosomes. But the non-invasive prenatal test was abnormal, so the woman was offered the opportunity to participate in our study. And what's interesting is she had a family history of cancer. So her father was diagnosed with Hodgkin lymphoma and her mother with melanoma. And this is what the sequencing results look like. So if you look on the x-axis here, this is chromosome 1, 2, 3, 4, 5, 6, 7, etc. And these are Z-scores. What you're seeing here are fragments of DNA that are sequenced and mapped to individual chromosomes, the short arm and the long arm. And what it should look like on a normal result is this orange line should be straight across. Well, even if you're not a geneticist, you can see that there's excess sequence from the short arm of chromosome 2. That's very abnormal. There's also excess in chromosome 5. There's excess in chromosome 9, and there is a deficient amount on chromosome 11. And as you go across, there are multiple aneuploidies in several chromosomes. This is highly suspicious for cancer. She had a whole body MRI at the NIH. And you can see here, I hope you can see my pointer, but you can see this mass that is white and it's bilobed. And here it is on the cross-sectional view. Now my screen has frozen. There we go. So this woman turned out to have Hodgkin lymphoma. So she was treated for that, even though, she was really not too interested in additional testing. She ended up having a diagnosis of cancer. and her baby with a cardiac abnormality and is a carrier for Fragile X syndrome. The second case is another 31-year-old woman in her first pregnancy in the second trimester who had non-reportable testing results 3 times and was also shown to have aneuploidies, multiple aneuploidies. Again, felt very well, essentially nothing too exciting in the past medical or the family history. Now, if you look at her results, she's got everything looking pretty normal here until you get to chromosome 14, where there's monosomy 14, and it looks like monosomy 22 and monosomy X. So again, multiple aneuploidies, not as abnormal looking as the prior case, but still quite abnormal looking. And this woman on the MRI from the NIH turns out to have a massive fibroid. Here's the baby. The baby's head is down. Here's the baby's body. And this fibroid, it's almost like having twins. The fibroid is as big as the baby's body. So what's happening here is the fibroid is undergoing apoptosis as well, or in the previous case, the tumor is undergoing apoptosis. And in both cases, they're shedding DNA, excess amounts of DNA into the maternal circulation, and that is throwing off the ratios. In both cases, the fetus is essentially fine. In this case, fetus was perfectly normal. In the other case, the baby had an unrelated cardiac anomaly. So thus far, I've been trying to show you that over really just a decade, there's been a rapid transformation of prenatal testing that it's really unprecedented. And part of the reason for that, as I've already alluded to, is that industry played a role in disrupting technology. So you're all familiar with disruptive technology, how Uber really disrupted the taxi industry. Google Maps took over regular maps. I mean, I've got two sons who don't know how to read a map. Airbnb disrupted the hotel industry, and now we have prenatal DNA testing. Well, I've kind of emphasized mostly the benefits of the testing, but there are some not so good aspects to it. For one thing, there was recently an article that was published by Reuters that suggested that one of the very big providers in China, the Beijing Genome Institute, is actually using routine prenatal testing to get DNA from millions of pregnant women. Now, they truly have done millions of tests. And this what they're talking about is data that was published in an article in the very prestigious journal Cell, where because they've had so many of these cases in their commercial lab, they can use it on a research basis. I don't know whether they are actually consenting these women or not, but they can anonymously look for findings. And for example, they showed in this article that the sequencing data actually revealed the presence of different viral infections that had become incorporated into the maternal DNA. They also showed ethnic origin. Now, these women are not consenting to this analysis of their ethnic origin. And if you look at the lower left box there, these are different ethnic populations within China. They also showed specific loci that were predictive of maternal height, weight, and fertility, as well as predisposition to twin gestations. So there's been some controversy about this as to, this unintended use of this testing, and it is kind of the dark side of it. In addition, as I've highlighted in the title of my talk, there are other consequences with regard to gender reveal parties, which I didn't even know about until I started reading about them. And this ability to get a blood test that's highly accurate to determine the sex of your baby has resulted in a multi-million-dollar industry in gender reveal parties. And here's a woman who has her eyes closed and somebody is setting off fireworks in the background with pink smoke indicating that she's going to have a little girl. Wonderful. Except that there have now been reports of people getting killed. There was also a major fire that was started by an explosion in, I believe it was in Arizona or in California during a drought. And that led to hundreds of thousands of dollars of damage that, in fact, the family had to make some sort of reparations. But several people have now been killed by flying shrapnel at gender reveal parties. And then one of my colleagues in the Netherlands, Jan Oosterwijk, he made a joke and said, NIPT really stands for non-intentional pre-symptomatic tumor diagnosis. As I said, it functions well as a crude liquid biopsy, even though it wasn't designed for this. We're of the philosophy that we're actually helping these women because we're detecting these cancers pre-symptomatically, and they're getting treated once it's detected. But there are other people who might say that we're not doing, that these women didn't ask to be diagnosed with cancer, and that maybe we're not doing such a good thing. We are creating anxiety, we are limiting the wonderful experience of their pregnancies. So I also put it in the category of unintended consequences. So I have some additional thoughts. Just I'm going to wrap up now. I see there are some questions in the chat box, which is great. Things that make this field unusual, as I've already alluded to, include the fact that industry, rather than academia, has driven innovation. Of course, academia has played a part, but for most other genetic tests, they were started in academia, maybe validated through clinical trials, federally funded clinical trials, and then licensed to commercial laboratories. In this case, the commercial laboratories really accelerated innovation. A lot of the tests have been introduced before clinical utility has been established. I mentioned Jacobson syndrome. I don't think anybody would say that that is a public health problem and that we need to screen for that. It's being screened because we have the technical capability of doing it. I'm not going to rationalize why companies are doing it. This expanding test menu and the tests are just always moving ahead and more is being tested for. It's completely changed the paradigm for prenatal testing. And then in the pre-test counseling, a lot of times women are unaware that their DNA is being sequenced and analyzed as well. So this has increased the complexity. And in my former life, as a prenatal genomicist, it actually really did increase the counseling time. So my last slide is really just a summary that the cell-free trophoblast DNA serves as the proxy for the fetus in screening for aneuploidy. And it's becoming the first tier test because of this very high positive predictive value compared to the low predictive value of the prior tests, which resulted in a lot of unnecessary amniocenteses. The cell-free DNA sequencing has provided us with knowledge of previously hidden aneuploidies. We didn't know that perfectly normal women, for example, could have mosaic trisomy 8. We also are learning from rats or rare autosomal trisomies and confined placental mosaicism, how that can be correlated with abnormalities in fetal growth, for example. And we're increasingly aware of the fact that there are many cancers that are just present and are not causing any symptoms, and they can be detected using this technique. And again, what we're trying to do is get the evidence to be able to advise clinicians what to do next, because really that evidence is needed. So I want to acknowledge the many people who have contributed to this work. The identified team at the NIH, as I've said, is multi-institute. Meredith Hawks-Miller and Mark Pertile were our collaborators on the rare autosomal trisomy study. I do have a laboratory at NHGRI. This is a picture that's a little bit old because it was pre-pandemic. We have not been able to have a lab meeting in person because of restrictions due to the pandemic. So I'm going to end there and I will stop sharing my screen and hopefully look at the chat box or the question and answer.
Thank you very much, Glenn.
Thank you.
So I'll go ahead and read out some of the questions to you then in the Q&A. But first, I'm going to start with my own question. I'll take a little liberty if I might. So I didn't really appreciate until you explained it in the in the bloodborne DNA that it's so overwhelmed by the mother's DNA to outcompete that of the placenta and the fetus. So it got me thinking, what about in cases of microchimerisms where previous pregnancies cells are in the mom? Do they shed DNA at a level that might be picked up and mistakenly attributed to a previous pregnancy?
So obviously, if you've done your homework, so some of our other work involves intact cells that persist from a prior pregnancy. But we've shown both in the mouse model and in humans that those cells actually migrate into the maternal organs. And there aren't that many of them. So even if they do shed DNA, that doesn't skew the results. So that is generally not a problem with this type of testing.
Okay, fair enough. Okay, let me just go to some of the questions here. So Anthony Lamonte asks, can fetal DNA and maternal blood be probed selectively for certain mutations such as triplet repeat expansions, et cetera?
Yeah, so some of the, so let me just say with single gene disorders, I didn't talk about that, but that's being increasingly able to be tested, especially for certain things like a skeletal dysplasia. If you're looking for something that's uniquely fetal, like achondroplasia, you can do that. With triplet repeats, the mother may be a carrier if it's inherited on the X chromosome. So you have the difficulty of distinguishing between the maternal and the placental DNA, and that's a lot trickier. And to my knowledge, no one has done that yet. So the recommended testing now would be to test the mother to understand if the fetus is at risk. Okay.
And then a follow-up from Anthony. He was asking about copy number variations beyond the most common ones, including like 22q11.2 or 16p 11.2. Can you detect those in fetal DNA and maternal blood?
Yes. So I didn't point out in some of the variation on the sequencing results, there were like single line spikes. Those are actually small, benign familial copy number variations. So you can detect copy number variations. It's more likely to be from the mother, but it could be from the fetus as well. It's pretty sensitive for copy number variations, at least that are 7 megabytes or larger, megabases or larger.
Okay. And then the follow-up question, how do you integrate development of technologies for liquid biopsy, increasingly sensitive DNA for non-fetal diagnostic evaluation versus that for targeted fetal diagnosis?
Can you say that again?
Yeah, he says, Anthony asked about integrating development of technologies for liquid biopsy that are increasingly sensitive for non-fetal diagnostic evaluation versus that you're targeting for fetal diagnosis. So I think he's relating this back to what you're saying, such as about the cancers, for example, that sort of thing. And
yeah, so there are a number of companies that are exclusively in the liquid biopsy field to detect cancer, and they're not in the prenatal field. A lot of your sensitivity will depend on the depth of your sequencing. So if you're doing shallow sequencing, you may not be able to have the sensitivity that you need to detect some of these variations. But if you're doing deeper sequencing, and as the technology becomes less and less expensive, you can afford to do deeper sequencing, then you are going to pick up more and more variation. And I should point out that there are significant price differences in some of the technology. And there's a third technology that I didn't even get into, which involves microarrays, and they don't detect the cancer at all. And they're very, very targeted. They're just looking for 13, 18, 21, for example.
Okay, and that's 3 times late. So that's one last question. One of our graduate students, Hassan Farah, asked a kind of a broad, high-level question, asked how child health, human development research has evolved or changed through your tenure as director at NICHD. What are the big changes, big picture you've seen?
Thank you for that question. So again, it's only been 5 years, but the biggest change is that that $1.6 billion that is our institute budget was previously funding many, many grants, but only 1 or 2 grants on a particular topic. There was no depth. So we underwent a strategic planning process in 2018 and 2019, where we decided what are our priorities? Where do we really want to go deep? And where do we want to partner with other institutes or other federal partners? So that's one thing. I mean, obviously, COVID has been a major emphasis for us for the last 2 years, and really advocating for children. In the beginning, people thought, oh, children they're not sick. Why should we worry about them? And then we learned about multi-system inflammatory syndrome in children. Almost 1,000 children have died. There was reluctance, understandably, to vaccinate children. Children were out of school. There will be long-term effects on development of that year where kids have been out of school. And we've also had to advocate for pregnant women who couldn't get vaccines in the beginning. And there was a lot of mixed messaging, even though we held a task force in 2017 and 2018, making recommendations on how to do research on pregnant people involving vaccines or therapeutics. So I think the biggest thing is that really trying to focus our populations. Our third population are people with intellectual and physical disabilities who have also been somewhat forgotten during the pandemic. So really identifying within the NIH ecosystem who we are, who we represent, what are our goals, and that's true even before the pandemic, but we really had to ramp it up with the pandemic because Congress did not give us targeted funds. They gave some of the other institutes targeted funds. So we had to compete for funds that were given to Dr. Collins's office, and our fabulous teams really worked hard to understand who's at risk for this severe multi-system inflammatory syndrome in children, and also to develop ways of testing children in school to keep children in school, and especially children with intellectual disabilities who can't wear masks or they can't wash their hands, don't understand why you're putting a swab up their nose. So I kind of say that the 5 years have been like 2 different experiences, the first 3 years and then the second 2 years.
Yeah, I think I understand that completely. Well, look, I want to please everybody join me in thanking Dr. Bianchi for a fantastic presentation.
Thank you.
Thanks everybody for attending. And Danny, I'll see you in about 20 minutes.
Okay, I have the other link.